จกรรมในภาคบ่ายเราจะบรรยายเป็นภาษาอังกฤษนะครับในหัวข้อ Sustainable Agriculture for Good Health นะครับ There are three professors. The first professor is Dr. Peter Sarnow. Let me introduce him a little bit. He is a professor in microbiology and immunology, Stanford University of Medicine, Stanford University, United States of America. He got elected to be the member of National Academy of Science in 2020. Today, he will present on the topic of role of non-coding RNAs in viral infection. Please welcome Dr. Peter Sarnow. Please give a big hand for him.
Thank you very much, Dr. Boonchue. I really want to thank the organizing committee for the wonderful invitation. And I would like to talk to you today about non-coding RNAs during viral infection. In particular, I want to talk to you about circular RNAs. Well, what we do know is there are two circular RNA viruses that can infect in cells. First of all, there are plant viruses. They're called plant viroids. These are tiny, naked circular RNAs that can infect plant cells, and they kill the plant. And they also can go from one plant to the other plant. The other thing we know, hepatitis B virus, which infects your liver, has a companion that's called hepatitis delta virus. Hepatitis delta virus also has a circular RNA molecule. And what it does, hepatitis delta virus steals all the capsid and matrix proteins from hepatitis B virus to amplify its RNA genome. So the question is, where does this circular RNA come from? And what has been known since 2006 by a paper in Science, that when you look at the RNA structure based of hepatitis delta virus, you see very much it folds into a ribozyme that looks like an RNA sequence that is encoded in a human mRNA encoding the CEPB protein. And what you can see, all these interactions are very, very conserved. Now, what was exciting about this finding, we know hepatitis B virus only infects human tissues, but not hepatitis B virus can also infect non-human mammalian tissue culture cells, but not hepatitis D virus. So that means basically hepatitis delta virus must have arisen 200 million years ago when humans suddenly split and emerged from other mammalian and non-mammalian species. Well, we asked the question, do circular RNAs also exist in human cells? And when you look by EM, electron microscopy, for a long time we have known already since 1979 there are circular RNAs in human cells. And some of them have tiny little knobs. That doesn't mean these are proteins, that means these are tiny little base pair interactions in circular RNAs. So they actually are in human cells. The question is, are they really there in huge amounts? And we knew this finding that the answer is yes since 2012, when Julia Salzman, when she was in the lab at Patrick Brown at Stanford University, found that many hundreds and thousands of circular RNAs are made in human cells. They're cell type specific expressed. They are expressed from 5 to 20% of transcribed genes. They are 1 to 10% abundant, and there are about 20,000 circular RNAs in a single human cell. The copy number is about 90% of circular RNAs. They only present 1 to 10 molecules per cell. So they're in small amounts, but if they're ribozymes, they could have catalytic activities and could have important function. So we asked, well, first of all, how are circular RNAs made? They're made by a backsplicing mechanism. What you see here is a DNA molecule that is being transcribed in human cells by RNA polymerase II. Here you have exons shown in a big bar, and here you have intron sequences. If the intron sequences can base pair with each other and bridge together by proteins, what can happen? The 5' branch point of the exon can attack the 3' splice donor, and basically you form a circular RNA molecule. Now, what we know so far, and that was very exciting for us, circular RNAs are made in the nucleus, as I just told you, by a back splicing mechanism. We know they come out in the cytoplasm. They interact with small RNAs, like microRNAs. They can interact with long non-coding RNAs. They also bind proteins, of course, and some of them get secreted, very much like the plant viroid enzyme from cell to cell. Some can be translated by ribosomes. So we asked the question, what happens when you infect cells with different viruses? And we have infected them with hepatitis C virus that also infects the liver, enterovirus 70A. Enteroviruses, as you know, is a very important virus in Asia, and also SARS coronavirus. And I want to tell you so far what we have learned so far from these studies. The first question was, well, what happens if you simply infect cells with hepatitis C virus? Does the cellular landscape change during the infected cells? And this study was done by a postdoctoral fellow from Taiwan, Shusei, together with Pakpoom, Boonchue, and Kunlaya Sumbunvivat. Both, of course, as you know, are very famous virologists from Thailand. And what we find, indeed, what we find is that when you look, you find that some circular RNAs are increased in abundance and some are decreased in abundance. And what we find is that circular RNAs are proviral. They're very, very important for hepatitis C virus. Hepatitis C virus is shown here. It's a plus-stranded RNA molecule. At the 5' end, it has a sequence element. It's called iris. That's very much like a prokaryotic Shine-Delgado sequence to which ribosomes can bind directly, very much like in bacteria. And we know that 170 million people are infected worldwide. We don't have a vaccine, but we have a wonderful compound called sofosbuvir. Sofosbuvir inhibits replication of the viral RNA, so we can get rid of the virus very easily after 12 weeks of treatment. But because there is no vaccine, if you get bad blood again, of course, then you get hepatitis C virus again. So when we had this data and we find these circular RNAs from the host cell are actually helping the virus to replicate, Pakpoom, when he was in my lab, asked the question, what about the circular RNAs, other circular RNA viruses? Are circular RNAs also being made from the viral RNA genome? And I never thought about this because if you think, I told you that the viral RNA is a single-stranded RNA molecule. If you make a circular RNA, you cleave it. So the viral RNA genome should be dead. So it doesn't make any sense. But the answer is it's kinetics. It's the kinetics. It doesn't happen early. It happens later in infection. And so what Pakpoom and Chen Kao, a postdoctoral fellow from China, what they find is, indeed, when you look whether circular RNAs are being made from the hepatitis C virus RNA genome, basically we are looking for sequences that are non-contiguous in sequence, and what they find, indeed, there are circular RNA made from all over the viral RNA genome. And what you see here, here's a 5' end and here's a 3' end, and there are clusters at the very 5' end throughout the genome, and there are clusters at the 3' end as well. Now, this is a prediction. Of course, we were very worried this could be cloning artifacts. This could be recombination events of RNA viruses, because the RNA-dependent RNA polymerase is error-prone. So what we needed to do, have a very stringent, a very solidified approach to test that these predicted circular RNAs are actually being made. So what we did is, and this is a key slide, and I will tell you what you should remember. Okay, we think we have a circular RNA made from hepatitis C virus. okay, is it real? So we simply anneal an oligonucleotide shown here, and then we add reverse transcriptase. If the circular RNA is indeed a circle, then RT will go and copy the circle many, many, many times, which you then can amplify by RT-PCR. So basically you have a repeated nucleotide chain, and then you can actually come in with primers A, B, and C, and basically if you have a repeated change, you see a fragment A, B, C, and D. And this tells you basically you have a bona fide circular RNA being made in infected cells. So for the remainder of my talk, whenever we see circles, just look whether we see A, B, C, and D, or we don't. And as you see here, basically we see some of the circles which I just showed you, they are being made, and here they're verified by sequencing, of course. The next question was, are they important for the virus? Well, what we knew is that one circular RNA actually could make a polyprotein. That's shown here. The sequence of the open reading frame is shown here in blue. Here's the start site, AUG codon. It should make a 20 amino acid small oligopeptide. Is this important? And the question is, yes, it is important, but how do you test it? You can test it by tagging this polypeptide. In this case, we took a tiny little tag. It's called a high-bit tag. And when we add a high-bit tag here, what we do find indeed that, as you can see here, over infection, the high-bit tag is being translated and we make a proviral peptide. Then the question is it the circle that helps the viral RNA, or basically is it simply the protein that's being made? So you can topically express the protein on its own. And when you do this and you look at the viral RNA, this is a nanoluciferase viral RNA, what you can see is if you express the protein, indeed you make more viral RNA. So it is a pro-viral RNA. So the big question was, how are these circular RNAs being generated? How are they being made? I told you splicing normally is important to make host cell RNA in the nucleus, but these RNA viruses do not go into the nucleus. They only stay in the cytoplasm. So the question is, well, if they are made in the cytoplasm, is basically viral translation important or viral replication? So what we can do is we inhibit translation. How do you inhibit translation? Well, there's a drug called cyclohexamide that inhibits translation, or we can introduce stop codon, so translation is not happening. So all you need to know is, and I told you, whenever you see these fragments A, B, C, and D, that means circulars are being made. So even if you inhibit by these two methods translation, you see circular RNAs are still being made. What about viral replication? In this case, again, we can use sofosbuvir. I told you that's a powerful compound that inhibits hepatitis C virus replication. Again, when you do this experiment, what you see is A, B, C, and D fragments are made. Again, even if you don't have replication, the circular RNAs are being made. So that's interesting. I told you in the nucleus, host cell circles are being made. In the cytoplasm, the RNA doesn't need to be translated, it doesn't need to be replicated, and there must be a spliceosome-independent mechanism. So in order to test how this works, we basically, what we did is we made tiny little single-stranded RNA molecules that we know could be circularized from the viral RNA genome. And so what we did is we have a linear fragment which contains flanking sequences. We contain fragments that only have the 5' or the 3' half of the same flanking sequence, or simply the bona fide circular RNA coding sequence on its own. And when we test this, what we find, what is very interesting, is that we do need both flanking sequences in order to make circular RNAs. shown here, basically. And again, you see A, B, C are being made. If you only express half of the circle, which has a 5' sequence or the 3' sequences, no ABCs are being made, no circles are being made. Now, where are they being made from? How does the nucleotide sequence looks like? And what we find is, for the most part, the circular RNAs are being made by cleavage of loop regions in the RNA. So when we had a closer look at these loop regions, what we found is there's a sequence, conserved sequence motif at the 5' end CUU and at the 3' end also CUU. So basically, these are cleavage sites for RNase L, ribonuclease L, and that cleaves RNA at these particular sites. So we had a hypothesis that maybe this particular nuclease is involved in cleaving the viral RNA. Of course, we also need to know what is a ligase that links the ends together, and I have to say we don't know yet. Okay, is RNase L being important for making these circles? And what we have two approaches. One, we have a compound. It's shown here. It's called valerianic acid dilactone. It inhibits RNase L. When you add this, no fragments ABC are being made, no circles are being made. So this nuclease, if you inhibit its activity, basically what you see is no circles are being made. The other thing is, of course, we can do a G knockout. We knock out RNase L in cells. And again, when we do this, shown here, RNase L minus, 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 you see knockout cells do not make these fragments ABC. No circles are being made. And as you see here in number 9 is a positive control where RNase L is being made. Okay, so in summary to this point, we know hepatitis C virus makes circular RNAs, some make novel proteins, and they are very important for the virus to enhance their function. And we know RNase L is important for making them. At the very end of my talk, I will tell you why are these circular RNAs so important. But what I want to tell you briefly, two things. If viral circular RNAs are important, the question is, are they also generated from other RNA viruses? It shouldn't be just important for hepatitis C virus. So one virus we start working with is enterovirus 70A. As I told you at the beginning, this virus is a picorna virus, also blood-stranded RNA virus, and it causes hand, foot, and mouth disease virus that it mostly affects children. And it also can cause neurological diseases. So it's a very important virus to study, and no vaccines are available for this virus. So when we looked, what we find is there is a circle being made, but very different from what I showed you with HCV. Remember, they were all over the viral RNA. In this case, it's only at this particular junction between a capsid protein and a viral protein called 2A, that's a viral protease. And what we find, that's very interesting, right at this junction where the circular RNA is being made, there is an RNA recombination hotspot. So our hypothesis is that the circle is important for generating variants and recombinant enterovirus 71s. And we are testing this right now. And again, of course, we wanted to know, is this circular RNA important for virus infection? So what we can do, here's a circular RNA, which we verified by this ABC rolling circle amplification. We can add an siRNA against the junction that should destroy the circle, and we can have a mutant siRNA that should not destroy the circle. And when we do this experiment, what you see here, basically this is the fold change over the empty vector. We see indeed the circular RNA, when you get rid of it with an siRNA against the junction, but not against the mutant siRNA, HCV levels drop. So again, it's very pro-viral. So finally, I have another 8 minutes or so, what I want to tell you, we started working on SARS-CoV-2 because everyone has been studying, and as you know, we have normal ongoing or current variants popping out all the time. We need to know how can we attack this virus. Are the vaccines we have good enough? And also, are there biomarkers? Because we have been so surprised being virologists. It's not just infecting your lung, as you know. It affects many, many organs in your body. And there's something like long COVID, which we never thought would happen with an RNA virus. It's not a retrovirus, not like HIV, but we really needed to study this in more detail. So we asked the question, does SARS-CoV-2 make circular RNAs? If so, are they important for the virus? What happens if we get rid of them? What you see here is, again, we did make libraries and we sequenced libraries, and what we find is, indeed, there are circular RNAs being made, and one is highly, highly abundant, and it's made in the plus and in the minus strand conformation from a very particular region of the viral RNA, and that's shown here. What you see here is the entire 30,000 nucleotide of SARS-CoV-2. As you probably remember, ORF1A and ORF1B, these are the enzymes that multiply, that amplify the viral RNA genome. These are the auxiliary proteins, and they are different between SARS and MERS and other coronaviruses. So dependent what you have here gives you either a SARS-CoV-2 or MERS virus, which, as you know, or many of you know, infects camels in the Middle East. So what we find is this circle I just showed you is about 1,400 nucleotides in length, and it's very highly abundant, and it's in the plus and in the minus strand conformation. And I will tell you in a minute what this TRS means. These are sites that are important for transcription and replication. And I will tell you in a second why we are super excited about this particular circle. Well, indeed, here are the data. When you look, we don't see A, B, C, and D because the circle is so big. In this case, it's 1,400 nucleotides in length, so we see only A. But we see it's accumulating over time in the plus and in the minus orientation. Now, in order to tell you why we're so excited about this, I need to remind you, and maybe not too many of you know about this, basically, what is the replication strategy of this virus, which is very, very unusual. Here's a viral RNA genome, okay? So you make your polymerase, ORF1A or ORF1B, and it binds at the very end. It binds and it copies. And you see these red boxes here. Whenever it hits the red box, and it's called transcription replication site B for body, it jumps to the very 5' end. Here it uses the second, third, fourth, and fifth, and it jumps to the 5' end. So these are subgenomic minus-stranded RNA, very unusual for RNA viruses. And we predict, of course, that the plus-stranded circle should bind to the minus-stranded RNA. What you see here, the minus-stranded RNA, of course, gets copied into mRNA molecules. And the mRNA molecules contain, in each case, the leader sequences, which is shown here at the very, very 5' end. So basically, the minus-stranded circle should bind and inhibit the leader sequence. So my point is this circle, which can bind to the minus and to the plus-stranded RNA, should be very, very powerful in modulating translation and transcription of the viral RNA genome. And we are testing this right now as we speak. It has been difficult so far to test it because we didn't have a recombinant genome we could manipulate, but now we can do it. And we can work with under facilities that are not BSL-3. So we can do this experiment right now. So the second question was, well, these are tissue culture experiments. Are these circular RNAs important? Can you find them in infected samples? So what you see here, We have obtained samples from pegylated interferon lambda treated outpatients at Stanford University, which had COVID-19. And basically what we obtained 10 samples, nasal swabs, and we asked basically, are these circular RNAs I just showed you present in these samples? And the answer is yes. So here you see the circle being present in some samples, not in every sample, but in some. The other thing you see also, if you have the same sample, basically stored for different lengths of time, when you cleave it with the RNase R nuclease, some are more sensitive than others. But clearly, the circular RNAs are in nasal samples. The next question was, what about all these variants? As you know, you always hear there's a new variant out there. And they are known as WA1. That's one of the first variants we had in the United States. It's the Washington strain. And then, as you know, we had Alpha, Omicron BA, Omicron BA.5, and so on, Alpha, Delta, and they all come out. And we tested as many as we could. And as you can see here, each one makes this particular circle. So we think this circle is very, very important for SARS-CoV-2, and it doesn't matter what variant you have. I told you it comes from a particular reason. So we can make a mutation in the viral genome, and indeed the circle gets smaller because we deleted this region. And so that tells us also we're looking at the right thing. So that's super interesting. As you know, many questions are, where does the virus come from? And we also know many of these viruses, not just SARS, Ebola virus, many other viruses hang out in bats. So we asked the question, is this virus made in bats, infected with SARS coronavirus? And the answer is yes. Here what we have done is we have used a particular bat cell. We expressed the receptor for SARS-CoV-2, infected them with SARS-CoV-2, and here is a circle. So it's not only important for the different variants, it's important also for its host restriction. Where is it? This might be a little bit hard to see. Where is this RNA located in infected cells? And probably what you can see here, the blue is the nucleus, and what's outside here is the cytoplasm. I apologize, it's hard to see here. We see it if we overexpress just in vitro synthesized circle, or we see it also here when we infect cells with SARS-CoV-2. It's in the cytoplasm, and that's where we think it should be if it's important for the virus, because that's where the virus replicates. Now, another question is, can these circular RNAs be targeted in an animal system? And we can target it with CRISPR-Cas13, and we do this in collaboration with Philip Santangelo at Emory University. You probably know there's not only Cas12, there's also Cas13 that attacks RNA molecules. So we can take Cas13 and guide RNAs and basically formulate them in a polymer. The polymer can then basically, via nebulization, insert it into small animals. And when we do this experiment with Phil Santangelo, HCV titers drop. So it's also important in an animal model, in this case, hamster mice and small piglets. To sum up, what are the final thoughts about my talk? I told you that what are the function of non-iris containing viral circular RNAs? We don't know. If you have an iris, you make a protein. The protein is proviral. The function of the non-iris containing circular RNAs, we don't know. How are they generated? As I told you, by a nuclease, a particular nuclease, which I didn't go into detail, is induced after you infect cells with many viruses. So the virus has absorbed. He uses this mechanism to make its own circular RNAs. And the final question I want to discuss with you, do these circular RNAs, do they bind other molecules? And is that important for pathogenesis? I told you there are thousands of these RNAs being made. I do not think they have 1,000 different functions. They must function in bulk. And what we think is going on, that's our working model. Basically, you make circular RNA shown here from the host and also from the virus, right? They're in the cytoplasm and they bind a protein called PKR. PKR protein, when it gets activated, induces the cell into an innate immune response. The cell knows, I'm infected, I commit suicide. The cell goes into apoptosis. And what we think is going on, these RNAs, and there's evidence from Ling Chen in Shanghai, that they can inhibit the activation of PKR. And this, of course, is the most important slide. And what you see here is the acknowledgement. Most of the work was done by Chen Cao, shown right here, together with Pakpoom when he was in the lab, and Quanting Liu, he works with Enterize 71. Elise Grossi-Soyster and Kriti Shah, he is Elise and he is Kriti. They worked both with SARS coronavirus. And I think I ran out of time. And thank you for attention, and I'm happy to ask questions when there's time. Thank you very much.
Thank you, Peter. We have time for one or two questions from the audience. So if you have a question, could you please raise your hand? Okay. Thank you, Sixi.
Thank you very much. Great talk. I'm interested in the case of COVID-19. So you showed the several types of the virus that have the level of the cyclization is different depending on the strain. So is that somehow related to the potential of expansion or the infection or some other types of phenotype?
Yes, I think the RNAs is more or less induced by different viruses. And I think that's probably is very important. Also, as I mentioned, if RNA cleaves the viral RNA is dead. So making the circles, there's already kills the virus. But if early on you slowly amplify the viral RNA genome, that's in abundance, and then the circular RNAs will not interfere with this and still have a pathogenic signature. That's our hypothesis.
I see. And also, may I ask one more question? So, in the case of COVID-19, you explained that the coding sequence of the 3' region is bind to, or complementary bind to the 5' end. So, how those sequence limitation or sequence arrangement is occur? How, do you have any idea to since
yes.
The 3' region is coding region, so
that's
somehow
right. Yes.
limitation.
Yeah, so he's referring to the TRS element because it's in the plus and the minus strand circle can bind to the plus and minus subgenomic RNAs. The interaction is not very well understood. It's a very small, I think it's 50 nucleotide base pair interaction. So my gut feeling is maybe there are protein also involved in stabilizing this interaction.
Okay, thank you very much.
Thank you, Dr. Sato and Peter. So could you please give a big hand for Peter? Thank you. Next speaker, we're going to change from virus RNA to be the rhizobium studies. So
yes, because actually the sustainable agriculture and good health is actually come from the good source of plant, right?
Also, yes. Okay, next speaker is Dr. Eric Giraud from Plant Health Institute of Montpellier, France. He will present on the topic of Brady Rhizobium friendly bacteria that help plants to thrive thanks to their unique abilities. Welcome, Eric.
สวัสดี Yes, I would like first to thank the organizers of this conference that are also my colleagues. It's a real pleasure for me to be here and to present our works on how Brady Rhizobium can promote plant health. In nature, the plant have to cope with an incredible population of microorganisms, including bacteria, fungi, virus, nematode. It is estimated that there are between 1 to 10 billion of microorganisms per gram of soil, representing several hundreds of thousands of different taxa. And so the plant health will be directly linked to their ability to manage, to cope with this antagonistic population of microorganisms. During my talk, I would like to speak about how a group of bacteria, the Brady Rhizobium, succeed to enter in symbiosis with legume plants and how they can promote the plant health. The nitrogen is an essential element for the plant growth. There is a paradox. Nitrogen is abundant in the atmosphere, but under the dinitrogen form, the plant cannot use it. Some plants, such as the legume plant, have found a way around this problem by associating with nitrogen-fixing bacteria, collectively called rhizobia. And this association led to the formation of a nodule in which the bacteria fix nitrogen for the plant benefit and receive in exchange a carbon and energetic source that comes from the sunlight thanks to the photosynthesis. And so, thanks to this symbiotic interaction, it is not necessary to add a nitrogen chemical compound in the soil for the growth of legume crop. Here it is an example of a soybean field with a plot inoculated with a Brady Rhizobium strain and a non-inoculate slot. And so we can clearly observe that there is a clear advantage for the plant growth of this symbiotic interaction. So the understanding of the molecular mechanism that permit the establishment of this symbiosis is very important for the development of a more sustainable and healthy agriculture. The term rhizobium refers, in fact, to a very large and diverse group of bacteria that can form a nodule with nitrogen-fixing nodule with legumes. And among them, the Brady Rhizobium genus are the most common. Brady Rhizobium means slow rhizobium because these bacteria grow very slowly. But as the Italian proverbs say, chi va piano va sano e va lontano, means those who go slowly stay healthy and go far. And it appears to be exactly the case for the Brady Rhizobium. A recent study which aimed to establish which are the predominant bacteria on soil have shown that the Brady Rhizobium strain are the most ubiquitous and abundant bacteria in the soil on Earth. In addition, the Brady Rhizobium are most probably the bacteria that are the most widely used in agriculture. More than 80% of the legume crop produced are in fact able to induce nitrogen-fixing nodules with Brady Rhizobium. And in addition, the Brady Rhizobia are known to nodulate the largest number of legume species worldwide. And so we can ask, why such predominancy, why such ecological success of the Brady Rhizobium on Earth? There are most probably several explanations, but one of the first explanations concerns the genome size. The Brady Rhizobium contains a genome that is around between 8 to 12 megabase, and this is among the largest bacterial genome. Here, this is a representation of the first Brady Rhizobium strains which have been sequenced by the group of Professor Minamisawa. This is the genome of the strain USDR 110, his name has changed. It is not Brady Rhizobium diazoefficient. And it appears, in fact, that the Brady Rhizobium have acquired by lateral gene transfer a lot of genes, and by neofunctionalization, by duplication, in fact, they have created an incredible toolbox with a lot of sensor, regulator, transporter, and metabolic enzyme that enable them to adapt to very different conditions. The second possible explanation of the ecological success of the Brady Rhizobium is maybe found at the level of the structure of the outer membrane. It has been shown by the group of Antonio Molinaro that the Brady Rhizobium contains an opanoid molecule that is covalently attached to the lipid A of the lipopolysaccharide. And so this opanoid, in fact, act as a zip, and that permit to reinforce the stability and the rigidity of the two outer membrane, of the two outer leaflets layers that make out the outer membrane. We can observe here, for example, that the ultrastructure of a mutant strain that cannot produce opanoid is strongly altered in comparison to the wild-type strain. And it appears that the same mutant is also affected in its ability to cope with various abiotic stresses, such as acidity stress, or such as acid stress or detergent stress. In addition, the mutant can form nodules, but it cannot maintain a chronic infection because inside the nodules there are a lot of different biotic stress. And so the nodules age and degrade very, very rapidly. So it appears that these opanoids are key molecules for the Brady Rhizobia to maintain, to help them to cope with various stressful conditions, and which could explain maybe one of the reasons of the widespread dominance on Earth. Now, I would like to discuss about another unique property of the Brady Rhizobia. This is the diversity of the way that this bacteria can use to enter in symbiosis with legume. Normally, the establishment of the Rhizobium legume symbiosis involves a molecular dialogue between the two partners that permit them to recognize each other. First, the plant exudes in the soil some flavonoids compounds, and if these flavonoids are perceived by the bacteria, thanks to regulatory protein node D, this will activate the expression of some nodulation genes that we call node genes, and in return, the expression of these nodulation genes led to the synthesis and the secretion of a bacterial signal that are the node factor. And depending on the structure of this node factor, the plant can recognize or not the presence of the bacteria. And if it recognizes the presence of the bacteria, this will elicit a signaling pathway for which several actors have been identified, and that's led to the formation of the nodules and this concomitant infection by the bacteria. And so during a long time, it was assumed that all the Rhizobium legume symbiosis absolutely required the node factor for the establishment of the symbiosis. But in fact, it exists two alternative symbiotic pathways. The first one was described in some tropical legumes of the Ascinomene genus that have the unusual property to form root and stem nodules. The other very unique property of this symbiosis is that these bacteria, this plant, are nodulated by photosynthetic Brady Rhizobia that lack the canonical node ABC genes. Clearly, these bacteria cannot produce the node factors. And so, despite a lot of effort, we do not succeed to identify which are the signal molecules produced by photosynthetic Brady Rhizobia that triggers the nodulation process. But important progresses have been done at the plant level by my colleague Jean-François Arrigui. The screening of an EMS mutagenesis population of Ascinomene evenia has permitted to identify several mutant lines altered in the nodulation. And the identification of the mutated loci in the non-minus mutant line has permitted to show that several determinants of the node factor signaling pathway, here in green, are also recruited during these symbiotic processes, but not, as we could expect, the node factor receptor. In counterpart, this mutagenesis screen has permitted to identify two new symbiotic genes that both encode for receptor kinase and that we assume can sense the non-node factor signal that are produced by photosynthetic Brady Rhizobium. So we hope that thanks to the identification of this new putative sensor, it will be possible to identify this new symbiotic signaling pathway activating the nodulation in Ascinomene. The second node factor independent symbiotic process has been identified in soybean, a legume species that normally required the node factor for the triggering of the nodulation. However, it has been shown by the group of Shin Okazaki that the cultivar NREA can still be nodulated by a node-minus mutant of the strain USDR61. Very interestingly, in these last cases, this symbiosis is possible only if the bacteria possess a functional type 3 secretion system. If we mutated the type 3 secretion system, the nodulation is completely aborted. And so considering that most of the photosynthetic Brady Rhizobia also, besides lacking node genes, also lack the type 3 SS genes, this indicates that it exists at least two node factor independent symbiotic processes that differ by the requirement or not of the type 3 secretion system. So what is exactly the type 3 secretion system? This is a very complex nano-secretory machinery that looks like a nano-syringa. And this secretory machinery permits to inject directly into the host cells effector protein. And so the rhizobium effector most of the time act as pathogenic effector and can modulate the immune system of the host. And so depending on the host plant, it is observed that the type 3 SS can play a dual role. First, this type 3 SS can suppress the plant defense reaction and so promote the nodulation. We have here some data that illustrate some data obtained by Yi, that is here, Pongpangsong Watana. We observe here that the type 3 SS mutant of the strain RS3257 is less efficient on two legume crops, Vigna inguiculata and Vigna mongo. When we mutated the type 3 SS, the efficiency of the symbiosis is drastically reduced. So this indicates that the type 3 SS play a real positive role. But in the same time, on other plants, the type 3 SS can completely render incompatible the interaction. And so if we test the same strain on another legume plant, Vigna radiata, we can observe that the Y-type strain cannot nodulate. But the type 3 SS mutant is perfectly able to induce the formation of the nodules. In these last cases, this results to the fact that some effectors are perceived, some effector protein are perceived by some resistant protein that will then activate an effector triggering immunity response. This is an hypersensitive response during which the bacterial and the plant cells death. And so this permits to limit the bacterial infection. So these two examples clearly illustrate that the type 3 SS can play a dual role, a positive or negative role, depending on the host plant. But the data obtained by Shin Okazaki also, in these last cases, we have shown that it is only one effector that is responsible of this incompatibility. this effector is NOP2. When we mutate in NOP2, we restore a perfect nodulation. And so the data obtained by Shin Okazaki indicate also that some effector can directly activate the nodulation program and bypass the requirement of a node factor signaling pathway. So interestingly, it has been shown also that the strain USDR61 is able to nodulate Ascinomene, the first legume species that I spoke. And we can observe here that this strain is able to induce nodules. And we observe also that a lot of different non-photosynthetic strain in green can also induce nodules. And among them, there is a strain ORS3257 that appears the most efficient. This strain, in contrast to the strain USDR61, the strain USDR61 only induce nodules which are infected intercellularly, while the strain ORS3257 can induce nodules which are infected intracellularly and for which we can detect nitrogenase activity. When we mutated the node genes, we do not impact on the symbiosis, but if we mutated the type 3SS, we completely abolish the nodulation, confirming that in these last cases, the process do not depend on the node factor, but depend on the type 3 secretion system. And so to progress in the understanding of which effector are required during this process, we focus on the strain ORS37 as a model. And so to make short a long story, it could be possible, thanks to the second genome, to predict which are the putative effectors that are translocated by the type 3 secretion system machinery. And by mutating one by one these different effectors, it could be found the one that play an important role during this symbiotic process. And like that, we have shown that, in fact, this symbiotic process relies on a cocktail of 5 effectors that play complementary and synergistic role. We observe that the mutation of the two effectors, NOP-M and NOP-P, result to the formation of nodules which are necrotic, indicating that they are plant defense reaction. And so this indicates that these two effectors, NOP-M and NOP-P, permit to repress the plant immune system. In addition, we have shown that the mutation in the two effectors, NOP-T and NOP-B, result to the formation of nodules which are non-infected, indicating that these two effectors permit the intracellular infection of the nodules. And finally, we identify a completely new effector that we name RNA for effector required for nodulation. When this effector, when the genes encoding this effector is mutated, the nodulation is completely aborted. Very interestingly, the RNA, we identify RNA homologue in around 2/3 of the strain containing a type 3 secretion system. And if we mutated RNA in another strain, the strain USDR61, we observe that the mutant lost its ability to nodulate, confirming the general role played by RNA in Brady Rhizobium to induce nodule formation. In addition, if we transfer RNA in the strain D19 that contain a type 3 secretion system but that lack RNA, we confer to the strain the ability to nodulate. We have a gain of function in nodulation. In addition, we have shown that when we express RNA in transgenic area root line of Ascinomene, the formation of nodule-like structure, all along the root system in the absence of the bacteria. So all this data indicate that RNA alone is able to trigger the nodulation program in legumes. Nowadays, we have identified 6 distinct effectors from different bacteria that are able to induce a nodulation in either Ascinomene or in soybean glycine max. These distinct effectors, we propose to gather them in a new family that we call ET node for effector triggering nodulation. And so our main topic now is to try to understand how this ET node activated the nodulation program in legumes. What is exactly the mechanism? So to conclude, it appears that the Brady Rhizobia are the most widespread and abundant bacteria on Earth. And these bacteria can can promote the plant health, especially in plants that grow in soil that are poor in nitrogen. What makes unique the Brady Rhizobia is the diversity of the way they can use to enter in symbiosis with legumes. It exists, in fact, 3 distinct pathways. A common pathway that it is found in most of the Rhizobium legume symbiosis that involves the node factors. And we have shown that this symbiotic process can be modulated positively or negatively by the type-free secretion system. It exists a second node pathway, which is independent of the node factor and of the type-free secretion system. We don't know yet which are the signal molecules that trigger the nodulation program in these cases, but we have identified some putative receptor of this signal. And finally, it exists a third signal, process that do not depend on the node genes, but relies on the type-free secretion system and on a new class of effector that we call ET node that are able to trigger the nodulation program and that act in synergy with other effectors that are necessary to repress the plant immune system and to promote the infection. So the diversity of the way the Brady Rhizobia are able to enter in symbiosis with legumes make this bacteria very promising for future agricultural application. I think that a major challenge in the future will be to transfer this nitrogen-fixing symbiosis to cereal that remains the main food for the human. And so we can hope that by exploring new symbiotic pathways that apparently are more simpler, it could open to new promising opportunity to transfer more easily this nitrogen-fixing symbiosis to cereal. I would like to thank you for your attention, and I would like to sincerely express my gratitude to all my colleagues that have been involved in this project, in particular the SUT team with whom we collaborate since more than 10 years together. Many thanks for your attention.
Thank you, Dr. Eric. So we have 1 or 2 questions from the floor. So if you have a question, oh, Peter.
I have a question. Are there effectors that are triggering nodulation in these different pathways? Are they redundant or do they function under different conditions?
Yes, this is a very important question, in particular for the type-free system, because all the strains that contain a type-free secretion system contain also node genes. And so the main question is to know if there are specific conditions for one pathway in comparison to the other, or if the type-free secretion system acts to reinforce the ability of the node factor to trigger nodulation. This is a question for which we don't have yet answer, but yes, it's sure that there is a strong overlap in the type-free secretion system and on the node factor dependent pathway.
Thank you very much. Great talk. I'm curious about the host specificity of the ET node effector. So when you introduce your ET node effector to soybean, do you have the formation of the nodule?
So for the moment, we have mainly worked on RNA and on ascinomyelin. What I know is that Shin-Okazaki tried to overexpress not RNA BUL25. This is another etinode effector in soybean. And apparently this is not so clear if the effector alone in soybean is able to induce nodulation formation. On our side, we have tried to transfer RNA in soybean. It doesn't work, but we are not a specialist on the genetic of soybean, so I'm not confident about this data.
Thank you very much. So I think we give a big hand to Professor Eric Giraud again. Thank you. Yes. Okay. And the last speaker for our session is Professor Dr. Shusei Sato from Tohoku University, Japan. And his topic is the evolutionary study of root nodule symbiosis toward applications for sustainable agriculture. So, Professor Sato, please.
Please welcome.
Okay, thank you very much for kind introduction, and thank you for inviting me to this great symposium. That's my probably fourth or fifth time to visit SUT in 3 years. So I'm happy to have to stand here again. Okay, so let me continue to the topic of root nodule. So since this session is sustainable agriculture, so from my talk, so I'd like to change the viewpoint from the plant side and put some evolutionary viewpoint for how the legume plant establish this fine system. So as Eric already mentioned, we have a lot of nitrogen in the atmosphere. And as Eric said, the organisms that cannot use the aerial nitrogen. So basically, we depend on the nitrogen fixation by bacteria. But fortunately or unfortunately, the human being have the potential to fix nitrogen and use the aerial nitrogen, make the ammonia and provide to plant. So, but that causes severe damage to the globe. So do you know the planetary boundaries that somehow explain the limitation of our universe? And there are 2 points. Surpass the limitation. One is genetic diversity, and the other is biochemical flows, especially for nitrogen. So we put too much nitrogen created by human. That breaks our limitation or planetary limitation. So considering the sustainable agriculture, we need to think about reduce the usage of human fixed nitrogen or the chemical fertilizer. So one of the natural based and ecologically friendly solution to reduce the nitrogen fertilizer is root nodule symbiosis, as Eric mentioned. So this is also Eric mentioned. The potential of the symbiosis between legume plant and rhizobia is extremely high. So if we grow the Lotus japonicus, one of the model legumes, in the pot without containing nitrogen, we cannot grow the plant. But if we add the rhizobium to the pot, we can get flower and seed. So the rhizobium provide enough nitrogen source to the legume plant. So this is also mentioned by Eric. This is the majority of the legume plant has invite or recognize what the signal exchange between rhizobium and legume plant occurs on the surface of root hair. Since this system brings the bacteria into the cell, so strict limitation for the bacteria recognition of the partner is very important. So as Eric mentioned, the flavonoid and node factor signal exchange is one of the key points of the partner conformation. So this occurs, this occurs on the surface of root hair. Then, if the host plant, legume plant, recognize this rhizobia as a partner, they start creation of the organ, nodule organ, and also they bring the bacteria into the nodule, guided by the infection threat. But in this phase, the bacteria is still outside of the cell, Then, and also the partner selection or partner conformation is ongoing in this infection threat. Then the final test finished. That goes, infection threat leads to the nodule cell. And then bacteria release into the nodule cell. So in this stage, the intracellular infection of rhizobia has completed. Then rhizobia start fix nitrogen. So inside nodule cell, you can see the infected cell and uninfected cells. So you can see the difference of the size due to the growth or increase of the number of bacteria inside. So inside nodule cell, bacteria was surrounded by the membrane coming from the plant cell membrane and cause the specific organ-like structure called symbiosis. So this is the very special interaction, having the bacteria inside the cell. And this bacteria fix nitrogen, then provide the product to the legume plant. So this is very useful system. So it was found in the late 19th century. And soon after the discovery of this symbiosis, the scientists tried to transfer this system to the crop, other plant. But since then, many scientists tried, as Eric said, but no one had succeeded. Even recently, the Bill Gates Foundation put the big money to transfer this nodule symbiosis to maize or barley to save Africa. But this project is not, goal has been achieved. That's simply because this root nodule symbiosis has very complicated system. At least more than 100 genes are involved in this process. And so it is difficult to simply transfer those genes into other crop is hard to achieve. But if we expand the viewpoint of root nodule symbiosis, create the nodule-like structure and accommodate the nitrogen fixation bacteria, that can be found. Not only the legume plant, but also the some plants from some plants from Fagalaceae, Rosaraceae, and Cucumbitaceae. So, but this symbiotic interaction is not intracellular symbiosis. That's still intercellular. The bacteria is remaining outside of the cell, but still create the nodule and accommodate the bacteria to fix nitrogen. So if we consider this as the root nodule symbiosis, the process of root nodule symbiosis traces back to the common origin of those 4 plant clade. And recently, we have the strong achievement of the DNA sequencing. So we can do the comparative genome analysis of the plant species with having those root nodule symbiosis. So we can conclude there is the establishment of root nodule, basis of root nodule symbiosis is common ancestor of these 4 clade. So we consider this evolutionary viewpoint into our new project called reconstruction of nitrogen fixing symbiosis. So as I say, it is difficult to transfer the root nodule symbiosis to the crop such as wheat and barley. But if we set the target to the plant of this nitrogen-fixing clade plant in this nitrogen-fixing clade, even the plant which it has lost the ability of root nodule formation, but still they have the history that created nodule and accommodate nitrogen fixation bacteria. So if we set the target to the plant in the nitrogen fixation clade, we can approach the transfer of nitrogen fixation system to not introduce, but reconstruct. So since they lost the ability, so reduce or reconstruct the genes that lost the function. So that could be more simple than think about transfer the other plant. So, sorry, in this. So this project, we have set 4 sub-projects with the specialists of each project and set the target of to the strawberry, which was belong to the rosette. Since we have the quite high quality genome information of strawberry in our hand. So in the first group, we carry out the detailed comparative genome analysis between the plant with root nodule symbiosis in the nitrogen fixation clade and lost the plant which lost the symbiotic ability and found 2 key genes, NIN and RPG, are commonly lost among the plant which lost the symbiotic ability. So in the sub-project 3, we create the core vector which brings RPG and NIN as the commonly introduced to this project. And in sub-project 2, we check the functional, the remaining of the gene, or the genes which lost the function from the viewpoint of the gene function. So in this analysis, we introduce the several key genes which regulate the cascade of root nodule symbiosis in the Lotus japonicus into Lotus japonicus and also in strawberry. Then check the response of the downstream genes. So as we expected, some portion of genes are response in the strawberry, same as Lotus japonicus. But some groups of genes does not respond in strawberry. So we think the gene regulatory cascade is also somehow disrupted in the strawberry. And we checked those genes and found there are 3 transcription factors among these genes. So we considered if we introduce these 3 transcription factors, we could reconstruct the gene cascade of those remaining genes and test it. So we add 3 transcription factors with RPG and NIN and CCAM-K, activated form of CCAM-K, and introduce those 6 genes into the root of strawberry. And as we expected, we can obtain the nodule-like structure in the root of strawberry. So we could reconstruct the accommodate or root nodule-like structure in the strawberry. Then we check whether the rhizobium can accommodate it into that nodule-like structure. So we tested several rhizobium strains, which has the relatively wide range of the host, and inoculated to the strawberry root. So surprisingly, those rhizobium can go inside the strawberry root. And that was not found in the tomato and rice. So accommodating the rhizobium strains into the inside the root, still that is in between the cell, intercellular. But endophytic activity can be found against those strains. So whether we can reconstruct nitrogen fixation into the nodule-like structure. So we test inoculation of the plant, which transformed with empty vector and introduced the 6 genes. So when we inoculate the rhizobium, the plant with empty vector does not recover the growth. However, if we introduce the 6 genes and create the nodule-like structure, when we inoculate the rhizobium strain, we can gain the growth. So that means, and actually we can detect the nitrogen fixation. Interestingly, that those rhizobium strains just introduce into the strawberry root, also fix some sort of, can do the nitrogen fixation. But if we add the key genes, the potential is increased, and the phenotype is dramatically changed. So plant grows better, and the runner, which indicate the sufficient nitrogen, can be found. So by introducing 6 key genes, we can reconstruct nodule-like structure, and which can accommodate the rhizobium and fix nitrogen and provide some sort of nitrogen to the strawberry. So we could achieve the reconstruction. But still, this interaction is intercellular nodulation. And the amount of fixed nitrogen is limited. So then we check the phylogenetic tree again. So we have gained the root nodule formation and accommodate nitrogen fixation, the ancestor of these 4 clades. But except for the legume plant, the plant in the other clade lost their ability, since the efficiency of nitrogen fixation or this symbiosis is not enough to pay the not cost effective. So the difference is intercellular or intracellular. So next step we need to consider is how the legume plant establish the intracellular symbiosis. And clue of this goal came from the different project. So we analyzed in the sanction mechanism of the legume plant, which is, as we said before, there's the signal exchange between the legume plant and rhizobium to confirm the partner which the legume plant accommodate or not. But during that confirmation, legume plant does not check the ability of nitrogen fixation. So it is easy. So legume plant does not care about the nitrogen fixation ability when they bring the rhizobium into the cell. So when we create the strain which does not have nitrogen fixation, but still have the old signal need to show the partner, legume plant accommodate or brings this rhizobia into the cell. But they recognize those rhizobia and stop the growth or maturation of nodule and kill the rhizobium. So we want to know the mechanism, molecular mechanism of this sanction system and isolate the mutant named PINK4. So in which even the non-nitrogen fixation bacteria, we call them cheetah, can survive into the nodule. Okay, so we check the phenotype in the electron microscopy. So normally, non-fixing rhizobia will rise inside the nodule cell, but in PINK4, they still remain the symbiosis zone. So by using this mutant, we check what genes are upregulated when we inoculate the cheating non-fixing rhizobia. So we obtained the result that the hydrolase activity and membrane transport and oxidative signal or the peroxidase activity are upregulated depending on the PINK4. So those genes, by this result, we guess the sanction against cheating bacteria is somehow related to pathogenesis. So phagocytosis is the endocytosis against the bacteria, so which the membrane around the bacteria and brings them inside the cell and make them, by fusing the lysosome, they lyse the bacteria. So the phenotype against cheating bacteria is somehow resemble the phagocytosis. Then we isolate the gene which causes this mutation. And unfortunately, the identified gene does not have functional information. That was annotated as unknown. But surprisingly, we thought that this sanction system is legume-specific, but this gene has conserved not only legume, but also the angiosperm. But we check the expression level of this gene. So when we inoculate the cheating bacteria, this expression is very high. And if we inoculate the functional bacteria, the expression of this gene is reduced. But surprisingly, that gene is expressed in uninoculated route. So this gene is somehow repressed when the functional bacteria come. So this was confirmed by using the promoter fused to the gas. So the expression of this gene was upregulated both cheating bacteria and functional bacteria in the nodule primordia. So that was not released into the nodule cell. Then when it was functional, this expression was reduced, suppressed. So this was upregulated in the regardless of nitrogen fixation ability and is suppressed when infected by the functional cell. So in that case, if we continue to express this gene, even the honest functional rhizobium cannot create the nodule. And the answer is yes. If we introduce this gene by the constitutive promoter, if we inoculate the wild type rhizobium, they cannot create the nodule. Okay? So from those results, we think how the legume plant established the intracellular symbiosis. So at first we isolate the PINK4 gene for the sanction mechanism, but this gene was expressed in root and it's suppressed when honest rhizobia, functional nitrogen-fixing bacteria, is released into the cell. So from those results, the legume plant may have established the intracellular symbiosis by acquiring the ability to recognize the nitrogen-fixing rhizobia and halt the lysis process by reducing the PINK4 gene. So next step we need to answer is how this pink4 gene was suppressed when the honest bacteria come. So we are now targeting the key genes controlling this expression and try to introduce these genes into the reconstructing bacteria. Then we try to reconstruct intracellular symbiosis in the strawberry. Okay, sorry for the taking too time. So this, by thanking those collaborators, I finish my talk.
Thank you, Dr. Sato. Yes, okay. Because the limited of time, so may I invite Professor Dr. Ng Tiamrung on the stage to give the token appreciation to the presenter. So let's start with Dr. Sato.
Yes. Thank you very much.
This is a very interesting topic. So if you have any question, we can talk later with him in a break.
Oh, on the stage,ครับ. And Peter and Eric also. So let's start with Dr. Sato. Dr. Peter. And Dr. Eric Giraud.
ครับและเรียนเชิญท่านรองศาสตราจารย์ดร.พันลดาติตตบุตรด้วยนะครับเรียนเชิญครับท่าน Moderator ของเรานะครับและท่านรองศาสตราจารย์ดร.ภาคภูมิบุญชื่นครับ Moderator ของเรานะครับท่านเป็นอาจารย์สาขาวิชาเทคโนโลยีชีวภาพจากสำนักวิชาเทคโนโลยีการเกษตรครับมหาวิทยาลัยเทคโนโลยีสุรนารีครับ